Hello, boys and the girl. Good morning. This is this video is meant for class 11th biology students, and this is for 11th September 2021. As we have been discussing in live classes about the plasma membrane in eukaryotic cells, now I'm right now going to cell organelle 2, that is cell wall. As all of you know, cell wall is the outermost membrane in case of plant cell, and there will be no cell wall in case of animal cell. So it is a non-living rigid structure found outer to the plasma membrane of fungi as well as in plants. What is it made up of? Cell wall of algae is made up of cellulose, galactans, mannans, and minerals like calcium carbonate, CaCO3. In other plants, it consists of cellulose, hemicellulose, pectins, and proteins. Now you must remember these chemical substances. Cellulose is a carbohydrate, hemicellulose, pectins, and proteins. Cell wall of young plant cell, primary wall, is capable of growth. It gradually diminishes as the cell matures, and the secondary wall is formed on the inner side, that is towards the plasma membrane. So basically it is like this. So the outermost one, and you can see when we take the section, there will be layers like you got middle lamella, primary cell wall, plasma membrane, and you can see the presence of pectin. Pectin is very rough and tough substance, and there is cross-linking glycan and cellulose microfibrils. So these are the chemicals that are present in the cell wall, right? And the middle lamellae, we were talking of the middle lamellae on top, is a layer mainly of calcium pectate. So middle lamella is made up of calcium pectate. It adheres, glues neighboring cells together. So it is the main function of this middle lamella is to adjoin the cells. The cell wall and middle lamellae may be traversed by plasma desmata, which connect the cytoplasm of neighboring cells. So there will be cross walls called plasma desmata. They connect the cytoplasm of neighboring cells. So cell to cell connection will be there. So what are the functions of cell wall? It gives shape to the cell as you have been seeing it. It protects the cell from mechanical damage and infection. It helps in cell to cell interaction like middle lamellae we said, plasma desmata we said, and it acts as barrier to undesirable macromolecules. Something unwanted materials, small particles try to enter inside the cell. This becomes a guard. It will protect. It won't allow those undesirable elements to get inside. These are the functions of cell wall. And then we go to endomembrane system, means there will be cell organelles which will be having their own membrane. We said in prokaryotic, membraneless cell organelles. Here we are talking of all the cell organelles in eukaryotic will have their own plasma membranes. So it's a group of membranous organelles having coordinated functions. They include ER, endoplasmic reticulum. Look at that endoplasmic reticulum. It is connecting almost nuclear membrane to plasma membrane. It's a bridge, connecting bridge, just like a railway bridge. And Golgi complex, okay, you can see the presence of Golgi apparatus or Golgi complex, that parrot green color. And lysosomes, you can see those round, spherical round ones, and vacuoles. So all these are having endomembrane system. They have got that the connections. And already I told you in that endoplasmic reticulum there will be two types, S ER, smooth endoplasmic reticulum. As you are seeing, there will not be any ribosomes on that. On the other hand, if ribosomes are present on those tubules, then we call it as RER, rough endoplasmic reticulum. So ER is endoplasmic reticulum is of two types, rough endoplasmic reticulum RER, smooth endoplasmic reticulum SER. RER on their surface will have ribosomes. SER will not have any ribosomes. And I said lysosomes, normally you get in your examination, UPSC ND examination, these collect all the waste materials, go to the surface of the cell and blast themselves. So they die while doing the function. So we say, since they are doing ultimate sacrifice, we say atmahatya, suicidal bags of the cell, lysosomes. Then endoplasmic reticulum, you can see clearly here, rough endoplasmic reticulum and smooth endoplasmic reticulum. As I said, it's like a railway bridge connecting nuclear membrane with the cell membrane. So RER or endoplasmic reticulum in general, these are a network of tiny tubular structures scattered in the cytoplasm. Endoplasmic reticulum divides the intracellular space into two compartments, as you are seeing. Inside endoplasmic reticulum and outside extraluminal cytoplasm. So it is two types as we discussed. One is the rough one having ribosomes on the surface. Another is the smooth without any ribosomes. And rough endoplasmic reticulum, it bears ribosomes on the surface. RER is frequently observed in the cells actively involved in protein synthesis and secretion. This is very important. Many times the function of RER is asked. Protein synthesis and secretion of enzymes. These are extensive and continuous with the outer membrane of the nucleus. On the other hand, SER, obviously ribosomes will not be there. They are absent. It is a major site for synthesis of lipids. Look at that. RER is mainly protein synthesis. SER is mainly for lipid synthesis. Lipids means fats. So in animal cells, lipid-like steroidal hormones are synthesized in SER. This you must remember. This is very, very important. And then densely stained reticular structures near nucleus is Golgi apparatus. It is named as Golgi apparatus. It is named after the scientist who discovered it, Golgi, right? Camillo Golgi, way back 1898. He observed first. So this organelle is appropriately called as Golgi apparatus. And as you are seeing in the diagram, there will be two structures together present. One is the vesicles, round structures, okay, bubble-like structures. Another is the tube-like structures, cisternae. Look at that. And cisternae form these vesicles. So you got two phases for this Golgi apparatus, cis phase and trans phase. So Golgi apparatus consists of flat disc-shaped sacs, bag-like structures. Sac means bag-like structures, cisternae, measuring 0.5 to 1 nanometer diameter. These are stacked parallel to each other. Look at that. They are situated one above the other. Cisternae are concentrically arranged with convex, that's why cis forming phase, and concave trans maturing phase. So one is the younger one, cis phase, and trans phase is the matured one. Cis and trans phase are totally different, but they are interconnected as you are seeing it. Okay, there is lumen also in the center. What are the functions of Golgi apparatus? It secretes materials to the intracellular targets or outside the cell. Materials to be packaged as vesicles from endoplasmic reticulum fuse with the cis phase and move towards the trans phase. This is why Golgi apparatus remains in close association with endoplasmic reticulum. And proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. So basically it is the conversions of proteins. That is the main thing. And it helps in the formation of glycoproteins and glycolipids. Along with that, of course, Golgi apparatus is known for throwing out waste materials. Membrane bound, just now we said lysosomes. Next, membrane bound vascular structures formed by the process of packaging in the Golgi apparatus. So lysosomal vesicles contain almost all types of enzymes, hydrolytic enzymes. So hydrolases, lipases, proteases, lipases, enzymes that digest lipids, proteases, enzymes that digest proteins, carbohydrates, enzymes that digest carbohydrates. They are active at the acidic pH. So that is the condition. They digest carbohydrates, proteins, lipids as well as nucleic acids. And I said most important one, they collect all the waste and commit suicide. So they blast themselves. That is lysosomes. And vacuoles, these are the membrane bound space found in the cytoplasm. It contains water, sap, okay, and excretory products and other materials not useful for the cell. Vacuole is bound by a single membrane called tonoplast. This is important. Many times in the exam this is asked. Tonoplast. Name the outer membrane of vacuole. Tonoplast. Remember. In plant cells, the vacuoles can occupy up to 90% of the volume of the cells. So, remaining cell organelles we will discuss in the live class. Until then, keep practicing.